So within this program that uh, this simple schematic representation that you are visualizing in the slide, that there is a sequence of amino acids which is in a particular file format, and that sequence of the amino acids or the protein sequence will be manipulated in the program that is called a modeler. And that program, after running that program, the output will be such like that you will be visualizing or analyzing a 3D structure of protein, and it will be taken into account for various experiments, for drug designing, for vaccine designing, for dynamic simulations, or for the analysis of the expression of various proteins. Everything that can be taken into account will be done through the 3D structure of a protein. And see, how to obtain that program. You simply go to the modeler of the Salil lab. This is the URL address of the modeler, and within that URL you will be visualizing the user interface of that website. And in that website, you choose for downloading the particular platform OS of that system you are having, and according to it you will be downloading the uh, this modeler, latest version of the modeler. You will be downloading it. Now, what are the how the modeler is working, how that modeler will create the structure of a protein, and what are the steps behind that developing the structure of a protein, that will be the presented here. Now, within that steps, there are certain files in a particular format, and these files or have some editing in that. You can take the files from the modeler website also, and after those files that have been included in the steps of the modeling of a protein, and there will be new output files also. So these are the files which are needed for for building a 3D structure of protein. And that alignment file will be there, profile .py, PDB comparison file will be there, compare script will be there, alignment script will be there, and modeling model development script will be there. The extension .py that you are visualizing here is the extension of the Python programming environment. When you are working with the modeler, make sure that your system is having Python working environment installed in that in your system. Then these extension files with the py, then they will be executed in your system. Otherwise, there will be error in execution of those file system. So uh, when you are starting the this programming of the designing a new model of the protein, then the files that are required, you are see that you will be creating a folder in the C drive, as the location of the every file that has been there in the modeling of protein is location is described in the C drive. And the model this model folder is there, in which these files have been put, and they have been run from there itself only. So the root directory that is present in that folder will be the root directory will be there, and the C directory will be considered for the modeling of the protein for those files. You will be choosing a particular target sequence of the protein that you have to find out. And is it visible? Parol should be.
So the molecule is like very highly
Yeah,
intense.
background is somewhat very highly resolution.
Yes,
Huh?
yes.
I will change. So let it be. I don't know how it. Let it be. It will take time, I think. So first of all, you have to choose the target sequence that will be and that will be taken into account for the formation of the structure. Then that target you will be able to find in the NCPI in the FASTA format and FASTA format. Then with the file name, you will be saving like this way that tvlds.ali or the target protein name will be there. .ali will be the extension, uh, and the sequence will be ended with the asterisk. This is called the PIR format, Protein Information Resource format, that will be used in the protein structure formation. So, alignment file will be created by the target sequence. Then the profile for this file will be there, that pdb underscore 95 protein information resource extension will be there, and that will be run by the build profile.py, and this will be the output files that will be generated with the target sequence, the alignment file that has been generated for the target sequence, and it will be run under the build profile, and the output files will be generated with the name of underscore 95 bin uh, with the build profile.prf and .ali. This is the script. This is the script of build profile, in which the highlighted uh, uh, or the edited uh, keywords are there, and these things are only the editing part. Any target protein you will be choosing, then these highlight change the or edit the keywords here, and run command will be somewhat like this, that C drive is the running drive, and the root directory will be of model build profile.py, and simply enter on the command prompt, and the file will be run, and the output files will be generated, and this is the result of the build profile. Then compare, and when the build profile is been done and you will be getting the output files, then you have to compare the target sequence with the template sequence. Every model that has been generated is not something like that. There is an algorithm, and through that algorithm it will straight away generate the various templates for the model of the protein. It is generating the models by comparing the predefined models that have been similar to its sequence. For that only the sequence is required to compare or building the profile with the template section. So these for comparison, the template should be selected from the PDB coordinates, and this will be run in this for file script or the modeler script, in which the templates are selected and with their chains, amino acid chains are there, and they will be mentioned in the later side of the this is the PDB ID of the template proteins. The template proteins are those proteins which are having sequence similarity much more closer to the target sequence for which you are creating or generating a model. So choosing these templates with their respective chains, and editing the required portion of the script for the comparison profile, you will be running this particular command on the modeler thing and generating the other part of the see the comparison result that will be there. The templates are there in with which this sequence is very much similar than that template. That one hrtb is the very much similar to the target sequence, and it will be taken into account for choosing a this 3D structure of that your target protein. Now, after uh, choosing the most similar protein, the alignment with that protein sequence will be made through the alignment file, in which the input files are the your target file and the most relatable protein sequence file that will be aligned, and the output files will be like this way: .ali and .pap files will be generated by after running the alignment file. So the script of the alignment file is there. Your template file will be edited in the highlighted section with the chain mentioned in the command sequence, and your this target file will be there in the highlight in this highlighted section, and the command for running the alignment file is this align2d.py. On the command prompt, you just run this command, and you will be finding the then the last step is of modeling. When the alignment file is done and you will be getting the output file through the alignment procedure, then number of models you are selecting, that models you are generating, 5 models you are generating, maximum is 8 to 10 models that you can generate. But in that what will happen when the number of models that are that have to be generated are more than 5, then the repetition of the no, generated models will be very much similar. That you can say that duplicate models can be generated with the number of models are more than 5. So we are just choosing 3 models that have to be generated or 5 models have to be generated. Then by editing the highlighted the commands or the highlighted keywords with our target sequence files of the generated with the previous commands, we simple run the model dot single dot py. This command, after generation, the models will be generated, and it will be having some dope score, and according to the dope score, you will be comparing the models, which model is the best suited for your work or taken into account for the good model or the best model. Then after the modeling, the protein structure has been developed. What are the things that you can do further? That you have to check the stability of the protein. Then these are the servers in which you will be analyzing the protein structures for their stability or the quality of that structure. uh, ProCheck is there, SAF server is there, Ramachandran plot is there, Dali server is there. So these are the servers in which you can, according to their properties, you can visualize and analyze the protein structure quality as well as the stability of that protein structure. So these things are there. uh, I use mostly SAF server, where the Ramachandran plot is such a way that you can analyze the amino acid aggregation for the stability of the protein of stability of the structure. So this was my presentation, and for the protein structure. So you had mostly the thing is that biggest domain is either sequencing in bioinformatics, or either the structure analysis or structure development in bioinformatics. So moreover, the major portion of the study is DNA, RNA, and protein. And apart from that, drugs, vaccines, and the chemistry part, in which the drug molecules are taken into account. These are the major domains of working with the bioinformatics, and it is the major criteria. You can visualize the various servers are there which are working for the genetic analysis, genomic analysis, and proteomic analysis, various servers are there for the which are acting as a repository of the structures of the protein as well as the sequences of the protein. And apart from that, new AI based servers are also coming to account for such as Rosetta for protein annotation work, and AlphaFold for structure prediction. So various techniques have been developed with the AI and the machine learning processes, and it is very useful, very informative, very robust. They are having robust algorithm they are having to work with. So this is no very feasible for